Okay, let's get started. So, hi everyone. My name is Derek. I'm a business development manager here at GenoBioTX. First, it's a pleasure to welcome everyone to today's webinar. We're excited to have everyone here together for today's talk, which is titled Harnessing the Power of the Cre-loxP System, Benefits, Challenges, and Applications. Before we get started, I just want to quickly introduce GenoBioTX for those who might not be familiar. At GenoBioTX, so we're committed to advancing biomedical research by providing innovative and high-quality animal models that help accelerate the development of next-generation therapeutics. Our goal is to help bridge the gap between basic research and clinical application, and we're excited to share today insights that can help you achieve that. So for today's session, we're going to cover the following topics. So first, our speaker will introduce the Cre-loxP system that allows for conditional gene knockout, knockdown, and overexpression. They'll then cover the history of genetic recombination systems and how they enable precise control of gene expression. And then finally, they'll present GenoBioTX's genetically modified models that use these methods. Also, at the end, we'll have a Q&A session, so feel free to drop any questions in the chat as we go along. I'm thrilled to introduce our expert speaker today. So today we have Dr. Andrei Golovko. Dr. Golovko received his Master's of Science in Microbiology at Moscow State University in 1992. He also has his MBA in Marketing from Rutgers, as well as his PhD from the Institute of Cell Biology and Genetic Engineering from Ukraine in 2003. Dr. Golovko has more than 29 years of experience in genetics, genomics, and comparative medicine and disease modeling, both in academic and industry environments, including generating and analyzing genetically modified models used to identify potential drug targets, including MPC 1L1, which is the target for Schering multi-billion-dollar drug Zieta. Dr. Golovko now acts as the Associate Director of Business Development here at GenoBioTX. And without further ado, let's get started. Dr. Golovko, the floor is yours.
Okay, thanks, Derek, for the introduction. And all I just want to add is that I personally have many years of experience designing, creating, and analyzing Cre-lox strains, and will be happy to provide my insights today. So my presentation will consist of several parts. First, I will introduce the Cre-loxP system. I will discuss the practical issues with the Cre-loxP mice, some considerations for constructing Cre strains, and like a brief information about GenoBioTX and what we can offer in this field. So the Cre-loxP system is the efficient recombinase that was derived from P1 bacteriophage. It was originally developed by Dr. Sauer and patented by DuPont in 1988, and was initially used for activating gene expression in mammalian cell lines. In 1994, the laboratories of Drs. Marth and Rajewski reported that the Cre-loxP system can be used for conditional gene targeting. And since then, the publications and the use of the system has dramatically increased. So Cre stands for the causes for recombination. It's a 38 kilodalton site-specific enzyme that can catalyze recombination between two LoxP sites. And LoxP stands for the locus for crossover in P1. It's a 34-base DNA sequence, and it has a directionality. It has a palindrome sequences flanked the core sequence, and it's long enough to reduce the likelihood of producing identical sequences in the mouse genome. So the most commonly used feature of the system is deletion. So LoxP sites, when they're placed in the same orientation flanking a certain genetic element, and when exposed to Cre, can excise this element, and this is how we create deletion. So this is a single action. It's irreversible event. If we place the LoxP sites in the opposite orientation, we can actually create inversion. So it's a less commonly used feature, but it's still it has some very good applications. So it's actually reversible since the LoxP sites are not altered in this case. But some modified LoxP sites can be used to create non-reversible events. Also, the translocation, when the LoxP sites are on different chromosomes, and they can use to exchange like parts of those chromosomes. So the irreversible inversion was actually used quite often in the conditional gene trap alleles like 15, 20 years ago. So for Cre-loxP system to work, the pairs of the LoxP sites need to be identical. So to make the system more flexible, more versatile, there were like multiple mutations and modifications were added to the LoxP sites. And for example, the LOX71 and LOX66 are used are mutant LoxP sites that are used to produce non-reversible inversions or translocations. So how does the system work? How does it apply in practice? So we can conditionally inactivate or activate gene expression in a tissue-specific or inducible manner. So another important term that we will use a lot in this presentation is the term floxed. It's basically a genomic element flanked by the LoxP sites. So there are several breeding steps that will be needed to produce a conditional knockout. A tissue, usually you will need to start with the tissue-specific Cre strain, and you will need to breed it with the homozygous or heterozygous floxed strain. So as a result of this first step breeding, you create a heterozygous flox strain, and another will have the allele with the tissue-specific Cre. The next step will be to actually use this double positive mouse and breed it with the regional homozygous or heterozygous flox strain, and this is how you create Cre positive and homozygous conditional knockout. That will, for example, in this case, the knockout will be in liver tissues. So for transgenic strain to be expressed conditionally, you will also need to use a Cre strain and breed it with the mouse that has a conditional expression of the gene of interest. So only one step is needed because you can produce the conditional overexpressing gene of interest by breeding with the Cre line. So transgenic strains usually have a flox stop element that prevents transcription, but when exposed to the Cre from the tissue-specific promoter, this will activate gene expression. The expression cassette can be either randomly inserted in the genome or targeted to a specific safe harbor region where the insertion is not going to create position effect or mutation with phenotype. There are several loci that are being used. ROSA26 is one of them, for example. It's a non-coding RNA that also has its own promoter. So actually, this locus can be targeted with the gene of interest inserted so that the ROSA26 promoter will drive the expression. H11 is another locus, but it doesn't have its own promoter. So when exposed to the Cre, this alleles will activate gene expression. So the transgene can be any sequence of interest. It could be coding region, non-coding region, shRNA, anything. The stop element is usually a flox poly A or expression cassette. So the Cre-loxP system became popular in single-cell lineage studies. The original version includes a large expression construct with several flux reporter cassettes carrying different pairs of mutant LoxP sites, which can be expressed in the random manner when exposed to the Cre, resulting in the deletion of the stop elements and activation of the specific reporters. So the more advanced and maybe another approach is actually use inversion function of the Cre-loxP system, as well as deletion that will also result in differential expression of various reporters. So this alleles are called rainbow, one or two, depending on the structure of the reporters, since they're very useful for tracing exons through the nervous system. So Cre-loxP system translocation function can be used to mimic or recreate certain conditions, certain human conditions. For example, the acute myeloid leukemia is actually a translocation event between two chromosomes where the genes called MLL and ENL are fused, and also the MLL and AF9 genes are fused through the translocation. So, and as you can see, the cancer, this mice develop leukemia phenotype and die at a certain age. So another major improvement on the Cre-loxP system was making it inducible by fusing the ligand binding region to the human estrogen receptor, ER, with Cre recombinase. The fusion protein Cre-ER is localized in the cytoplasm. Only upon estrogen induction, the fused Cre protein can dissociate from the anchor protein HSP90 and enter the nucleus, where the Cre can recognize the LoxP sites and will produce recombination. Now we can control the injection time of the estrogen, and we can activate time-specific regulation of the gene recombination. In order to avoid the interference by the endogenous estrogen, a point mutation was generated in the ligand binding region, thus allowing the Cre-ER to only respond to the induction by exogenous synthetic estrogens, such as tamoxifen or 4-OHT. Such fusion protein is named as Cre-ERT. Later, it was found that the 3-point mutations in the human ligand binding protein domain, Cre-ER2 was much more efficient, much more sensitive to the synthetic estrogens than the Cre-ERT, and that's where we have Cre-ERT2 system. Okay, in my experience working with the Cre-lox and talking to other PIs, I've heard complaints that the strains don't produce correct recombination or expected phenotype. So there could be many reasons for that. So it's very important to evaluate these strains before using them for your work. So the Cre lines need to be evaluated for recombination efficiency, expression specificity, and leakiness of the expression. And there are several steps to assess the new or the Cre line that you want to use in your work. And we will discuss those steps in more detail. First of all, you need to review the mouse origin, background information, and relevant published articles. There could be a lot of similar Cre strains. For example, there are multiple Cre strains with insulin gene promoters. that are actually that were created by independent labs, and the sizes of the insulin promoter were quite different. So you might expect like different results from all these strains. So it's very important to see which strains you really want to use, and which strains will produce the results of interest. A good place to start is MGI Cre portal. It's a Jackson database, because they have well-documented and systematized Cre strain information that includes expression data in embryonic and postnatal tissues. So, and it allows you to search the database by promoter or tissue-specific expression. It has a lot of interesting data and very useful. Once you selected and obtained the strain, you may need to confirm that it's the correct strain by strain-specific genotyping. For some strains, only generic Cre, for example, is available. But again, if you're not sure if you have the right strain, it's very important to use a gene-specific, strain-specific genotyping, not just a generic Cre PCR or PCR assays. So Cre expression can be validated at the DNA, RNA, and protein level after breeding with the flux gene of interest, or it can be analyzed by breeding with the reporter strain. However, keep in mind that the same Cre line may produce different recombination efficiencies in different flux alleles. It's influenced by the expression level of the Cre recombinase, as well as the gene location and the size of the flux region. Reporter strains are a good means to confirm that the Cre is expressed in the right types of the cells and allows to check for the potential expression in reproductive system. Because unexpected germline recombination that produces whole body knockouts may pose a significant problem in experiments and lead to unexpected results if not detected. Here is a study report showing that germline recombination found in many analyzed strains. And in my personal experience, we had several strains that produce recombination in the germline cells, where we would be basically lose the conditional feature of the system and would have a whole body knockouts in our colony, again, unexpectedly. So it creates a lot of issues down the road and produces incorrect results. The goal is to avoid permanent flux region deletion in the germline. So the designing and using comprehensive genotyping strategies is very important. Only genotyping for the flux allele may not be enough. Therefore, it's also important to genotype your mice for the flux del or knockout allele. Here's an example how the knockout allele was detected in reproductive system and passed to the germline. Another example of leaky expression in reproductive system that resulted in non-specific global expression of tomato in this case, and in the on the left, and it's Cre-lox stop lox tomato litter mate on the right. So, and unexpected recombination was found later by the presence of the 20 base pair band. To remedy the leakage issues, you may need to avoid breeding the Cre positive flux mice of specific sex with known reproductive system Cre expression. For example, if you know that your males will probably express the Cre in the semen, in the reproductive organs, you may need to not use those and use females instead, or vice versa. Again, I've seen the strains that would pass the Cre expression in germline, and I always had to deal with those when I worked with mice. At the same time, actually, the expression of Cre in the reproductive organs can be useful if you want to convert your strain from the conditional knockout into whole body. There are several strains that are with known expression in the reproductive system, such as DPPA3, DDX4 Cre, and ZIP3 Cre. Another issue to keep in mind is you won't be able to produce homozygous knockout if both Cre and FLOX genes are on the same chromosome. So you may need to avoid those if you really need the homozygous conditional knockouts, or you may need to redesign or actually create a new strain of Cre in this case, or use the FLOX allele to recreate this strain on the same chromosome. It's also common not to get Cre expression at all in newly produced strains. The only way to remedy it is probably to screen for the multiple breeders. So never use just a single breeder, single founder for your future use and for your future experiments, but try to use several founders and test expression of Cre in those until you find the one that works well. Also, it's important to test your Cre construct before using it in targeting in animals. Cre mice typically don't require homozygosity. So heterozygous Cre strains usually enough for the Cre to be effectively expressing and causing recombination. And homozygous Cre strains may also lead to unexpected phenotypes, especially if it's a targeted knock-in into like a certain mouse locus with known phenotype, or it could be a position effect that wasn't manifested in the heterozygous mice, but you may be able to see it in the homozygous mice. Also, it could be some toxicity issues could be related to the overexpression of the Cre protein and interaction of Cre with the endogenous genes that may also lead to the phenotypes in the Cre mice, just the Cre mice without the FLOX region. So, for example, if you're using a strong promoter in pay, it may lead to leaking Cre expression that's without tamoxifen, that I will probably address later. For example, CAG could be like a very strong promoter that actually lead to the leaky Cre ERT2 expression. So to detect the issues with the Cre ER leakage, you can actually use reporter FLOX reporter strains. For example, here as you can see, there's leaking tomato expression in the non-induced tissues that carry the CDH5 Cre and AI3 or AI14 strains. So, and last but not least, the configuration of the URL of your FLOX allele may play important part. So you need to design the URL so the LoxP is not, insertion of the LoxP doesn't interfere with the normal gene expression. When designing FLOX strain, keep in mind that you need to knock out an exon that will cause a frame shift. If frame shift is not possible, then you may need to consider knocking out critical domain. In both cases, we're actually talking about so-called critical exon. So you need to avoid modifications of the regulatory regions. So placing your LoxP sites in the upstream of the 5' UTR may not be a good idea because it may interfere with the promoter elements. Also, you may need to avoid affecting splice sites. The rule of thumb is to place your LoxP sites at least 20 base pairs upstream and downstream of your exon. Again, always a good idea to design your LoxP site inside the large introns. So, and let's talk about what we need to consider when constructing the Cre mice. So there are many things to consider when you want to choose Cre or Cre-ER. It really depends on your applications and intended use. So first of all, the location of the Cre or Cre-ER insertion, if it's targeted or random insertions, and if it's a targeted insertion, what kind of linkage are you going to use? There are other things to consider. Cre is not inducible, so knockout time is uncontrollable. Cre-ER is inducible, so you can do a time-specific knockout. Observed results from Cre and Cre-ER may originate from different effects, and I will talk about this later. Cre has a higher recombination efficiency than Cre-ER, and Cre-ER offers better specificity than Cre. Cre-ER might have issues with the leaky expression. And also Cre-ER can be used for the lineage tracing studies. So observed results from Cre and Cre-ER have different origins. With Cre, the timing of action is uncontrollable, so the gene knockout may occur in early development or outside of the intended experimental time window. Also, Cre action is irreversible. Once a cell is labeled, the daughter cells will also be labeled. So the effect of Cre results from the accumulation from the zygote stage all the way up to the observation point. So cells labeled by the Cre strategy all originate from the original one where the targeting occurred when the Cre started to express. And observed results from Cre-ER results usually are derived from the gene expression when the drug is administered. So the results from Cre-ER are directly dependent on the gene expression profile with the tamoxifen induction window. Basically here's the induction window where the recombination will occur, and usually at some point if you don't administer tamoxifen more, the metabolism will allow to clear the tamoxifen, and all the cells that will express Cre will be actually those that were activated during this time window. I mentioned that Cre efficiency is higher than Cre-ER, but there is a way to actually improve it. So the self-cleaved inducible Cre showed a significantly higher efficiency in recombination compared with the conventional Cre-ER. It's called as Cre-ER system. It's one of the ways to improve efficiency and at the same time have very high level of the recombination effect. And here's some data showing that the Cre-ER has a better specificity in the analyzed strains. So for knocking alleles, location of the Cre insertion needs to be considered. Let's say if you can insert either upstream in the 5' region in the like maybe ATG part of the first exon of the first coding exon, but keep in mind that this insertion will create a probably a knockout mutation of your gene of interest. So if your gene of interest needs to be expressed at least at the same levels, then you may need to consider the strategy 2, where you insert the Cre or Cre-RT in the 3' region, right downstream of the right before stop codon, and you can use some kind of a linker. So this way your gene of interest will be intact and your Cre-ER will be expressed at certain level. Maybe not the same as the your gene of interest, but at least it will be done in the tissue specific manner. Several types of linkers can be used to drive expression of both the target gene and Cre from the same locus. Iris is the ribosome entry site. It produces lower expression levels in the downstream CDS. And 2A is the protein break element that produces the same level, but it may add amino acids to the to both proteins. So really, depending on what kind of application and what kind of results you want to have, you might want to consider Iris or 2A. There are several other linkers that can be used as well, but these two are the most commonly used. And here are some examples of the leaky Cre-ER expression in some Cre strains, KRT14 and TUR-Cre. So, and that's more or less that I have to talk about the Cre-loxP system, and I would like to say a few words about GenoBioTXs. As Derek mentioned, we are a company with a deep history. We are more than 23 years in business. We currently have more than 10,000 strains in our repository, and over those 23 years, we produced more than 20,000 those strains. We have more than 500 genetically modified cell lines, and we have capacity to produce more than 4,000 strains per year. We actually have a big collection of the Cre and the reporter strain that can be used in combination with those Cre lines. We encourage you to search our database. It's actually, the website is here. It's Find Cre, and actually we systematize them by the what type of application they can be used. Also, when you search our database, you can see that we try to put some information about the cell type where this specific Cre is expressed, the strain name, and some other information. And as far as the fluorescent reporters, we also have a list of those. You can see them in the fluorescent section of our database. And in most of them will have some information and some data showing the expression information from those strains. And if you are really interested, you can actually see you can actually contact us, and we will be able to provide information about those strains. So our vision is to provide global researchers with professional, comprehensive, and integrated animal model services to create universal, simplified, and efficient approaches towards uncovering the mysteries of life. And we can create custom model services. We can create custom models. We can make knockouts, knock-ins, conditional knockouts. We can make like novel Cre lines, for example. We have a long collection of the catalog mice that can be used for research immediately. And we provide pharmacology and pharmacodynamic services. And we also can provide other supportive services in addition to what to those mentioned. So, and that's basically everything that I have. And thanks for your attention, and I'll be happy to answer any questions that you have.